This is Katie McHugh. I'm here at the World ADC Summit with Muctarr Sesay, and you're the VP of Process Development at Goodwin.
Yes. Yes, thank you very much, and it's good to be here.
Well, it's lovely to have you. So we are going to talk about process development and ADC processing and Goodwin's position in the ADC space. So that would be a good place to start. Would you like to tell me a bit more about your company?
Yes, I'll be happy to. Goodwin Biotechnology is a contract process development and manufacturing organization. We've been around for almost 20 years, and we're based in Fort Lauderdale area in South Florida. And basically, we are a fully integrated manufacturing organization. And by that I mean we can essentially take a client project from the early stages. And that will include taking your protein from the cDNA through to creating a recent cell line, stable cell line, and then subsequent to that we will create cell banking, and following cell banking then we will then develop the cell culture manufacturing process and also the purification to create the purified either recombinant protein or monoclonal antibodies. With respect to ADC, there are several approaches towards it. We can work with a particular client in terms of developing a ADC, antibody drug conjugate molecule. We can start from the basic proof of concept, whereby we will look at different conditions to achieve 3 or 4 potential conjugates, and then that will be evaluated by the client and of course by GBI also to select the most appropriate drug conjugate. That usually goes fairly fast. In a matter of a few weeks or so we can get 3 or 4 representatives, and then after that it's evaluated. Once it's been evaluated, again we're very transparent, we work very closely with the client. We will then select the best conjugate, and after that has been selected, then we'll go through a verification process, and then if everything looks good, then we go into preclinical animal toxicity studies, and then of course after that is completed, we will work with the client to prepare the CMC and other regulatory information that is needed to file the IND with the regulatory authorities. And of course we can continue to work with the client from after they've been awarded, after they've gone through the IND filing, to manufacture the ADC or the naked antibody for phase 1 and phase 2 clinical trials. One thing I'd like to point out is that usually when we start the optimization and development, we tend to work backwards on GBI. By that I mean we look more in terms of what will the molecule and the process look like when you get into phase 3. And so we work backwards to look at all the various scale-up parameters that will allow you to be able to reproducibly manufacture this material on a very large scale, and also at a commercial scale. Okay,
and so obviously there are a lot more companies getting into the ADC field right now, and a lot more companies maturing and getting products closer and closer through clinical development to the market. How has, in your experience, how has things changed from a regulatory perspective with what you need to supply to the regulators with regard to the paperwork and things like that?
I think what you need to demonstrate, obviously, from the regulatory standpoint, you're looking for the identity, the potency, safety, and also the purity. So those are the key requirements. But what tends to happen, in fact, having attended most of the presentations during this conference, you tend to get a Gaussian distribution in terms of population of the antibody-drug conjugates in terms of the ratio between the drug and the antibody. To me, I believe that the key there is consistency. When you develop a process, you don't try to zoom in on getting a 4:1 mole ratio, but you get a population. And so from the regulatory standpoint, as long as you can consistently able to achieve that Gaussian or Poisson distribution of the antibody-drug conjugate, then I think you should be fine. Because some people tend to spend time zooming on getting a very homogeneous conjugate. But really, the main point there is that if you get a heterogeneous conjugate, as long as you're consistently able to get it, I think you should be able to be okay with the regulatory aspects.
So if you're consistently heterogeneous, as long as the final product overall looks the same and that's reproducible.
Exactly, and of course being heterogeneous, but also it's efficacious.
Yeah.
And of course it's safe too, rather than, again, I'm looking at it more from the standpoint of the virtual companies and the small companies in which they don't have a whole lot of resources to put into it. But again, the key is really consistency from batch to batch.
Is that easy to achieve?
Well, much easier to achieve in terms of getting the population. What you do is you do as many batches as you can, and then you set up specifications. Typically, you will start a very wide range in terms of whether or not it's 2 to 4 to 1 of the drug to the antibody, and then you can narrow it down, and then you have to validate it. But the key again is that as long as you can demonstrate that you can do batch to batch using processes that we in part at GBI have actually developed a proprietary way of being able to modify the protein or the antibody that allows you then to get one molecule of the protein or the antibody, and then with various molecules of the drug. And so at the end of the day, as long as you can report you get 2 to 4 or 2 to 5, and you're able to do that consistently, you'll be fine. So I think after you've done quite a few of that, your confidence level should be high enough, and it shouldn't be that difficult to be able to achieve it.
And then what kind of clients do you have?
That's a very good question. I would say at GBI, we probably, I would say about 70% of our clients involves virtual companies, semi-virtual small companies, and maybe about 10% includes medical institutions, university medical institution, and the US government medical institutions. Then also maybe another 10% roughly, we're looking at large multinational drug companies.
Okay, and so there are a lot of people that are interested in getting into ADCs and kind of entering this field. And I think one thing that you'd want to find out is how do you actually partner up with a CMO? What does it entail, and how do you actually get your ADC made? Is it an easy process in the beginning?
Yeah, one thing, in fact, talking to quite a few people here during the last 2 or 3 days that I've been on this conference, is some people are here to learn, and I think with the advent of biosimilar, I think that ADC is well positioned to basically address the issue with not really biosimilar in itself, but bio-better. So if you imagine, basically you can use ADC technology of making an existing molecule that is coming off patent into a bio-better. And by that I mean, basically you increase the potency of the drug many fold, and essentially that will reduce your overall cost of goods, cost of manufacturing. And And I think the whole aspect again of doing ADCs is very complex. I mean, it's very complicated because you have to deal with not just the antibody and the tumor antigen target, but also you have to worry about the linker, and also you have to worry about the type of cytotoxic drugs. The challenge is going to be, because right now there's probably 2 or 3 major pharmas that have these very highly potent drugs. But for us, for GBI, what we tend to do is we'll work on less potent drugs initially to get a proof of concept. I think at the research level, small scale, it's easy to manipulate. Once you start getting into large scale bioprocesses, you're getting into the grams, multi-gram scale, then it becomes challenging from the standpoint of once you've created the drug conjugates, you got to figure out ways in a bioprocess environment to effectively remove side products and contaminants that you don't desire in the product. So in that regard, it takes quite a bit of effort, quite a bit of work, because there are certain processes that we use that will allow you to be able to linearly scale up the manufacturing from a few milligrams to kilogram scale. And that has come purely from a vast amount of experience with antibody itself, because we've been primarily a monoclonal antibody company over the past 20 years. And so we've gotten a whole lot of practice and experience in terms of handling antibody, naked antibody, and now moving into the antibody drug conjugate, we'll hone in on those experiences so that we can get an effective way to do the conjugation process, not just a small scale, but a large scale. And many of our clients that are coming are not well in tune to large scale processes, and that's where we're qualified to be able to address some of those challenges in terms of scalability, and then removal of residual contaminants, and most important, obviously, the quality of the product from the regulatory compliance standpoint.
Okay. And then how do you get your name out there? I mean, we were talking before about how it's quite competitive, the CMO space, with regards to antibody drug conjugates. How do you market yourself?
Conferences like this, and basically what we do is we target ourselves as not just an antibody cytotoxic drug company. We target ourselves more as a bioconjugation expertise organization. By that I mean, basically we have a wide array of clients that are not just looking for cytotoxic drugs, small molecule, but also radioimmunotherapy and radioimmunodiagnostics. It's also a very, fairly big area for us, and we've done quite a bit of work there, and quite a few of our conjugates are now in human clinical trials. And then also, in between, there are various other molecules that we can conjugate, like protein toxin. Some other factors, there was 2 or 3 presenters today that talked about, plant toxins. And we do have experience with that. And then, of course, we do have experience with dyes, fluorescent dyes. So for us, we position ourselves as a bioconjugation company of monoclonal antibody and recombinant proteins, using our vast experience over the past 10, 15 years or so in order to create a whole repertoire of antibody drug conjugates.
Okay. And can I just ask you as well, how have you found the conference? Have you met lots of people, or?
Yes, for me, being in the CMO business, we're a little bit restricted because we're client-driven. They come with sometimes partially developed processes or hardly developed, but they've done proof of concept. And so basically, I've attended pretty much the entire conference, and my education, has gone up significantly high because how the drug works, in vivo from the time basically it gets into the cell, the tumor cell internalize, and there are lysosomal degradations, and whether it's cleavable link or non-cleavable. This has been truly enlightening for me, and also I think the networking opportunity and the potential partnership that we hope to establish, I think here would be very, very valuable, particularly from GBI's standpoint, being a contract manufacturing. And also, of course, coming here, talking to various researchers, we're listening to them. I think we've gotten a pretty good idea as to the direction of ADCs in general, and I think it's going to be really exciting in the next few years. And we want to be at the so-called cuff of the wave in terms of this huge wave that we're beginning to see. And of course, with Seattle's new product that was launched in August, just gives it more credence in terms of that. I think we're now at the end of the beginning, in terms of antibody drug conjugates.